Today we're going to focus on heredity. At the bottom of this slide there are three pictures. On the left you have the sperm and the egg. You have the male gamete and the female gamete. The male gamete, the sperm, has the blue chromatids inside it. The female gamete has the red, and you can see when it becomes a fertilized egg, a zygote, you have one from dad, which is the blue, and one from mom, making a complete chromosome. And you can actually see, if we enlarge that and we look at the middle picture, you can actually see that there are different structures, okay, on each of these parts, on each of these chromatids. So you have the black for fur color, okay, black versus brown. And you can see black and brown fur color is an allele. And you can see the black and brown color allele is at a certain location, and those are the loci. If you go down to the bottom, you have long and short fur. So the long and short fur, that is at a different loci, a different location. Now the third picture is on the right-hand side, and that is a Punnett square. We're looking at the alleles from the female and the alleles from the male, and we're crossing the information from their genetics to make a possible offspring outcome. So let's talk more about Punnett squares. Punnett squares were named after R.C. Punnett. He's the one who designed and came up with this idea. It is a method to show genetic crosses. But the key here is that this is a probable outcome, what could happen. So if we get one child, one offspring, there are perhaps 4 possible outcomes that that child would have a certain genetic trait. So you can see the very bottom picture, there's a Punnett square, and basically if I have 2 parents that have a medium height, the possibility of one of their children could be tall height. A possibility of 2 of their children could be medium height, and a possibility of one of their children would be little t, short height. These are possibilities, not talking about 4 children, but talking about what that one child could have. Now on the right side of the slide, we're dealing with a monohybrid cross versus a dihybrid cross. A monohybrid cross only deals with one allele at a certain loci, only dealing with one locus, only one location. Remember, the prefix mono means one. Like bicycle, 2 wheels, mono meaning one. So a monohybrid cross matches one type of trait from the 2 parents. So if you look at the picture, the focus is color. Yellow seeds versus green seeds. That's only one trait, talking about seed color. But if you go down to the bottom where it says a dihybrid cross, it's actually crossing 2 traits, okay, which are on 2 different loci, 2 different locations of the chromosome. So the 2 traits here are not just color, but texture. So color is red. The color will be green and yellow, while the texture will be round, like smooth, versus wrinkled. And you can see that a dihybrid cross will not have 4 like the monohybrid cross, 4 squares, but it will have a total of 16 outcomes, possible outcomes. So here we have a dominant versus recessive. Here we have a Punnett square, and I'm going to focus on the terms dominant versus recessive. A dominant trait is the stronger trait, the one that shows up, while the recessive trait is the weaker trait. So here we have big B and big B. That would yield the dominant brown eyes. Now in this case, there is no middle trait. There is no hazel eyes in this example. So there would be no incomplete dominance, just regular dominance. So big B, little b, however, would be still dominant. So it would still give brown eyes. So another child, again, brown eyes. But then this last one, little b, will yield a recessive, a weaker trait, showing up blue eyes. So the 3 that have the dominant trait will all be brown eyes in this case, while there would only be 25% probability that the child would have blue eyes. Now, if we go to the focus on genotype versus phenotype, remember, genotype is the genetic description of the alleles. This is focusing on the big B, little b, and little b. This is focusing on the alleles on the chromosomes. So the focus on the chromosomes. While the phenotype is the physical expression. What does it give us physically? This is going to be the brown eyes, the blue eyes. In other cases, black here, blonde here. Okay, the size of our nose, if we have attached earlobes or unattached earlobes, and so on. Okay, when we mate them together in a Punnett square, the parents are going to be called the P1 generation. So the mother and father are going to be the P1 generation. Now the offspring, or the children that are yielded from the parents, would be the first generation or first filial generation. Filial means son or daughter. Then, if we have grandchildren, that's going to be called the second generation, or the F2 generation. Okay, let's talk about incomplete dominance and codominance. We said before that regular dominance, the homozygous dominant and the heterozygous dominant would all be dominant. We saw that in the previous slide. But here, incomplete dominance gives a mixed. So this is where you might have black here, brown here, and blonde here, or brown eyes, hazel eyes, and blue eyes. Here we have incomplete dominance. We have a red flower mated with a white flower, and that gives us a pink flower. So that big R, little r, that heterozygous, in the case of incomplete dominance, will be pink, a mixed color. But for codominance, it wouldn't be pink. It would actually still give a mix, but it would be, maybe with those flowers, it might be a red flower with white dots. In this case, the example of roan cattle, if we have a white cow and a orange colored cow, it will give you a white cow with orange spots. You also can see the flower there, in which it's pink, in which it's red with white spots. That is codominance. A great example of that would be blood types. Here you can see the difference between incomplete dominance and codominance. Here's another picture that illustrates the zygote giving the genetic information, specifically if it's going to yield a boy or a girl, the 23rd chromosome. And this picture here, we have a heterozygous black guinea pig, which is big B, little b, and a homozygous recessive brown guinea pig, little b. The black-furred father will be giving, in this case, randomly, either a big B or a little b. And you can see in the picture, it's going to be giving the genetic information of a big B, which would be for hair color. And the homozygous brown-furred guinea pig can only give a little b. So what do you think the children are going to be? Well, in this case, it's going to be a big B, little b.